Hello. Today I'll be talking about the cytoskeleton. The cytoskeleton is basically a bunch of fibers that help keep the structure of the cell. They help them just stay intact. So this is my cytoskeleton. These are my notes. No one is here. I'm in the study room by myself. And before we talk about eukaryotic cells and their cytoskeleton, when we look at prokaryotes, there are three components to a cytoskeleton in general. You have the microfilaments, you have the microtubules, and you have the intermediate filaments. When we look at prokaryotes, there is something called the PTZ, which is something that is equivalent to microtubules. They have the same function as microtubules, but for prokaryotes, it's FTDZ. And equivalent to the microfilament in a eukaryote, prokaryotes have something called MREB. And then when you look at intermediate filaments of eukaryotes, for prokaryotes, you have something called crescentin. Crescentin. Yeah, those are for prokaryotes. And that is something that I need to know for the exam. All right, like I said, the three components of the cytoskeleton are microtubules, microfilaments, and intermediate filaments. When we look at the microtubules, you have subunits. You have one subunit which consists of one alpha and one beta. And when you look at the microfilaments, they are made of 13 protofilaments. And each one of those protofilaments are made of tubulin proteins. And amongst the three components of our skeleton, they are the biggest ones. They are 25 nanometers. And the energy that they use is called GTP. And when you look at microfilaments, they're made of something called G-actin. Their monomers are of G-actin. They're 7 nanometers long, and their subunit is of one G-actin monomer. And one monomer, one sub, wait, their lengths, the little, yeah, they are the microfilaments. One microfilament is made of two chains of F-actin. Yeah. And then for intermediate filaments, they are made of keratin. Yeah, they are made of keratin. I'll get into more detail a bit later. And then so when you look at the structure of microtubules, you have the protofilament here, correct? You have one line here. And when you look at the monomers, you have beta, alpha, beta, alpha. They are alternating. The plus end of the protofilaments is called the beta end, and the negative end is called the alpha end. And so whenever you look at the structure of a filament, of the protofilaments, it's always going to be one positive and one negative. It'll never be negative, negative, positive, positive. And then when you look at, yeah, and that's usually called the plus minus end. That's basically what it is. And so when you're looking at the formation of the protofilaments, you are forming all 13 of them at the same time. And so you first start off with the one monomer unit, which consists of beta and alpha, which is called individual dimers. And then it goes to oligomers, yeah. And then once you start combining multiple part of those units together, you form something called the nucleus formation. Nucleus formation is basically when you're forming these filaments, once you are guaranteed that once you have more than two units together, monomers together, it's more guaranteed that the filament will grow longer. If it's just one unit, then it won't grow. It's not guaranteed as likely as if there were two or more units of the monomers together. That is nucleus formation. And this no nucleus, yeah, nucleus formation. And this is also both in the microfilament and the microtubules. And then there's also another term called treadmilling, which is basically apparent movement of subunits along a cytoskeleton element. They form plus to minus end, and they will keep adding and losing subunits. So the microfilament will always have plus to minus subunits because of the movement of these molecules. It's just going like that. It's going from the plus end to the minus end. Those monomers just simply keep moving. And that's treadmilling. It's like basically walking on tread. Yeah. And then when you look at the formation of the filaments, you are looking at I have to get some water. But when you look at the negative end of the microfilament, it has ADP actin, so low energy form. And then when you look at the positive end of the microfilament, it has the high energy form of actin. And so in these microfilaments, there's something called a critical concentration. It is basically the minimum amount of concentration that a microfilament needs to have. Excuse me. It's basically the concentration that the subunits in which the added subunits need to have. So say the critical concentration is 0.6, the added subunits need to have a actual concentration of higher. That's what the monomer's concentration needs to be. It needs to be a higher number. And that is basically what it is. And so when you're adding these monomers, you're adding it in the form of ATP G-actin. Yeah, and so you're first adding it to you're adding these subunits to the plus end. So the plus end has the ATP G-actin, and then once it's going down the microfilament, it's going to turn into ADP G-actin. And basically, the concentration, it basically controls the growth of the microtubules and where it grows in the microtubules. If they both have, yeah. So the whole point of the concentration is to be able to control how long these microfilaments can get. They could have a less concentration. These subunits can have a less concentration than the microfilament, and they won't need to grow because something is stopping it. And so when you're adding the GTP actin, there is something called a GTP cap. That's basically in the highest energy form, and it is seen added to the plus end of the microfilament. And then it goes down and it is released as GDP. And when it's released as GTP, it has a low concentration. Yeah. All right, another thing, there's something called yeah, so once you have a GTP tubulin added, these are not for, yeah, these are still microtubules, my bad. When you have the microfilaments, you have the GDP cap, and then once it's going down there, once it's going more down the actin, once you add the GTP, when you add it to the plus end of the microfilament of microtubules, you'll have a high concentration. That means a lot of subunits can be added. But once the GTP tubulin is hydrolyzed, then the GTP cap, as we talked earlier, it gets depleted. And then there's something called a catastrophe. That is where the subunits, they get loose. And in order to reclaim back a good level of concentration, you need to add a bunch of subunits with GTP, because remember the driving force, the energy driving force of microtubules is GDP. So you need to add subunits with a whole lot of GDP. And these are basically proteins that can stabilize or destabilize microtubules. So there is something called tau that can help stabilize subunits from the plus end. There's something called MAPS that will help to stabilize the plus end of the microtubule. And then there is something called MCAC, which is a catastrophin, which will increase the rate of catastrophes. All of these three proteins are used for microtubules. And so when we look at microtubules, Their function is to provide structure, and it is a railway for transport. So say a protein is added onto a microtubule, it'll be able to transport it to something else, to its designated sport place. And so one example is on the nerve cell. Say a motor protein of kinesin is added to the microtubule of an axon, it'll be able to transport it from the plus end to the minus end. Same thing with flagella. And on the cilia, you can add the microtubules. I mean, they could not add, they could support the body of the cilia and the flagella. When we look at cells, microtubules are used, they appear under the microfilaments. Microfilaments are at the top, but microtubules are the basis, the main form of the structure. And so when we look at flagella, there it is formed with 9 doublets and 2 singlets, meaning that in the center there are 2 sets of microtubules, and then on the outside there are 9 sets of two microtubules. So yeah, and so when you look at that, there is something called dyning, which will cause the microtubules to end, and that is basically what causes the flagella to move back and forth like that. It's basically called a whip-like motion. Now we move on to microfilaments. Microfilaments, their monomers are the G-actin. And in this case, they're called the ATP cap because they use ATP. And when you look at microfilaments, there is something called thymosin that helps to regulate how many available subunits there are for the microfilaments. Because if you keep adding more and more monomers of actin, it eventually might, implode or something like that. So with thymosin, it'll help to regulate how many subunits. And that also goes back to the nucleus formation. Once these monomers are getting added, then that is where formin comes in, introduced in microfilaments or in microtubules when enough monomers are attached to one another. Formin basically helps to stabilize the initial forming portion of the microfilament. It is more likely to lengthen. So same with the microfilaments and the microtubules. Formin helps with that. It helps to create that, it helps to drive that nucleus formation for the microfilaments. When you look at microfilaments, it also works the same. You have a negative end of ADP and a plus end of ATP. We're going to start with where the negative and the subunits, they get taken apart. They get taken apart by something called cofilin. They bind to low energy form of subunits and destabilize the microfilament. And when cofilin is attached, you now have, it's not able to go on the filament. So the monomer will get attached to cofilin. The cofilin will go away from GDP to from ADP to ATP. And then something called profilin will add on to the monomer so that it can start going onto the plus end of the actin, so of the microfilament. And so once the profilin is added onto the monomer, it'll be able to have to get added something called a thymosin as well, which is basically what we mentioned here. The thymosin will be able to regulate how many subunits can go on the plus end form. So once profilin is attached, they could use thymosin to kind of realize, oh, you can go, or you maybe cannot go. It can work, it can be attached, or it can get ripped away. But one key thing is the profilin is kind of like an indicator for thymosin to be like, oh, you can go or you cannot go, something like that. It's basically an indicator. It can get recycled. And then when the thymosin is added to the monomer, it can get to the plus end of the microfilament. And for microfilaments, To stabilize the plus end, there is something called the cap C. And for the minus end, there is something that can stabilize it called the tropimodulin. Yeah, stabilizing meaning there is no subunits being added or taken away. So cap C and tropimodulin will be able to take in charge of no subunits being added to each other, to the ends, I mean. Now, when we look at the structures of the microfilaments, they sit underneath the cell membrane, meaning that they determine the shape of the cell and of the crawling motion of the cell. And so it is shaped like this. This is an organism. It has a trailing edge and it has a leading edge. The leading edge is basically the direction the cell wants to travel. And then these are the leading projections, the little feet right there. And then there's something called the trailing edge. It is basically where you break down the microfilament and the membrane falls back to where there was support and drags the trailing end to that direction. And so that is the whole function of the microfilament. It gives the structure its shape and it'll be able to crawl. It'll be able to crawl, yeah. And then there are just some little molecules that we need to know that destabilize and stabilize the microtubules. So colchicine destabilizes microtubules. Taxol or paclitaxel stabilizes microtubules. Latrunculin destabilizes microfilaments. And then, oh my God, phalloidin stabilizes microfilaments. So destabilizing, meaning it will break away all of the subunits, and the stabilizes is basically when it will destabilize. Yeah, sorry. And then the structure of the intermediate filaments, what you have is something called a dimer. Dimer is basically two monomers that are intertwined together. One end has the amino acids labeled N, the other side is labeled carboxyl acids, carboxyl ends. And that is a dimer. When you take two of those dimers, they're called tetrametric protofilaments. And when you take two of these dimers, you can't have them combined with the same end next to each other. So if you have the amino acids on one end of one dimer, the next end, the dimer next to it has to have a carboxyl end on the other side. And then yeah, they can't have the same ends next to each other. And so when you look at this whole like non-matching of the intermediate filaments, that is basically called offsets. Offset, when the ends of the dimers of the intermediate filaments don't align, they don't match. And that is seen in something called protofilament assembly. So once you have this tetrametric protofilaments, you're going to take 4 sets of those and you're going to put them like that. 1, 2, 3, 4. And having that offset structure will enable you to align the ends together. And then once you have the protofilament assembly, you will have 8 of these protofilaments and they'll create one whole intermediate filament. And there are some functions of the intermediate filament. They are more stable for longer periods of time. They work in the nucleus to support nuclear membranes. They work in the cell junction. So example was the hemidesmosomes connecting to intermediate filaments. That's one example. And yeah, and that is basically the cytoskeleton. Thank you very much. Sorry for all my stuttering and have a wonderful day.